Welcome to this explainer. I am absolutely thrilled you're here. Today, we're taking a fascinating journey to demystify a subject that sounds incredibly intimidating at first, but is actually revolutionizing modern agriculture. We're talking about the world of lab-based cannabis science. For decades, the broader agricultural world has used high-tech methods to preserve valuable genetics and rapidly multiply elite plants. And today, well, this science is front and center in cannabis cultivation. So if you've ever felt completely overwhelmed by terms like micropropagation or aseptic technique, you are absolutely in the right place. We're going to break all of this down into clear, structured, and easy-to-understand concepts. I want to start off by asking you to imagine something. What if you could preserve a cannabis plant forever? And I don't just mean keeping a mother plant alive in a grow room for a few extra seasons, or hoarding a bunch of seeds in the back of your freezer. No, I'm talking about perfect, continuous preservation, completely protected from pests, protected from devastating diseases, and totally shielded from any kind of environmental stress. No way, right? Well, the answer actually isn't a traditional clone, and it's definitely not a seed. The secret is perfectly protected living tissue. A tiny piece of living plant growing inside a perfectly sterile container. This is tissue culture. And while it might sound like something ripped straight out of a futuristic medical laboratory rather than a standard grow room, I promise you, by the end of this explainer, we are going to translate all that heavy medical jargon into simple, highly accessible terms. So here is our roadmap for today. First, the tissue culture foundation. Second, explants and micropropagation. Third, controlling plant growth. Fourth, disease elimination and preservation. And finally, number 5, hardening off the plants. It's a logical, easy-to-follow journey through our ABCs. Let's get right to it. Okay, let's dive into this. Section 1, the tissue culture foundation, setting the stage for success. The A in our ABCs stands for aseptic technique. Aseptic simply means sterile. Now, out in nature, or even in a super clean greenhouse, a plant is naturally covered in bacteria, fungi, yeasts, countless microorganisms. And out there, most of them are totally harmless. But inside a tiny tissue culture vessel, the rules completely change. Even a microscopic, single contaminant can quickly take over the entire environment, completely overwhelming the plant. That's exactly why labs use specialized laminar flow hoods and strictly sterilized tools. And I love this realization. The plant often isn't the problem. The microbes are. It brilliantly demystifies early failures. A lot of beginners assume tissue culture is hard because the plant science itself is too complex. But in reality, most early failures happen because of contamination. Fungi and bacteria absolutely love the nutrient-rich gel we put these plants in, so the absolute name of the game is just keeping the bad guys out. Now, once we do have a perfectly sterile environment, that tiny piece of plant needs some food. Enter the B in our ABCs, basal media. In tissue culture, plants do not grow in soil. They don't grow in cocoa, rock wool, or even plain water. Instead, they grow on this specific culture media. Think of it as the ultimate sterile plant buffet that completely replaces the natural ground. The most common one used in cannabis tissue culture is called Murashige and Skoog media, or MS media for short. So what's actually in this buffet? Creating this plant food requires extreme precision. We're mixing essential minerals, macro and micronutrients, vitamins, sugar, water, plant growth regulators, and agar just to gel it all together. It's essentially a highly specific smoothie for a very demanding diva. Every single ingredient directly influences the plant's growth. Too much nutrient? Bam, you get toxicity. Too little? Development just grinds to a complete halt. Finding the exact right recipe for this complete plant meal is actually one of the biggest ongoing challenges for researchers. Let's transition into Section 2, explants and micropropagation, the building blocks of cloning. Now that our sterile stage is set, we can bring in the star of the show. This brings us to E for explants. An explant is the incredibly tiny starting piece of plant tissue that kicks off the entire process. And I mean tiny. Scientists use starting blocks no larger than a grain of rice. They rely on shoot tips, nodes, meristems, leaves, or stem sections. But here's the catch. Different cultivars behave very differently. Some produce shoots super easily, while others are incredibly stubborn or highly prone to contamination. A protocol that works perfectly for one variety might fail completely for another. And that leads us to C for cloning, or more accurately, micropropagation. People often think tissue culture is just another way to clone cannabis, and hey, they're partially right. But the scale is on another planet. When that tiny explant is placed on the sterile media under the right conditions, it develops new shoots. Those shoots can then be divided again and again. While traditional cloning might give you dozens of plants from a mother plant, micropropagation scales exponentially. You can potentially produce hundreds or literally thousands of genetically identical plants from a single donor. It completely blows traditional cloning numbers out of the water. Moving right along to Section 3, controlling plant growth, giving the chemical instructions. We have our perfectly sterile environment, our food, and our explant. But how do we tell that tiny piece of leaf or stem what we actually want it to turn into? Now, what's really interesting about this slide is comparing these two major growth regulators. Cytokinins encourage shoot formation and multiplication. On the flip side, auxins encourage rooting and cell expansion. They serve completely opposing yet totally complementary functions. So the absolutely crucial point here is that these growth regulators are literally the chemical instruction manual for the plant. By simply tweaking the exact ratio between those auxins and cytokinins, scientists can force the plant to make a specific choice. Make roots, make shoots, or just make an unorganized mass of callus tissue. Understanding exactly how to write these chemical instructions is one of the ultimate keys to successful tissue culture. Now we reach Section 4, Disease Elimination and Preservation, the superpowers of tissue culture. This is where tissue culture goes from being just a cool laboratory trick to an absolutely vital, industry-saving technology. Let's start with D for disease elimination. The cannabis industry is currently facing some massive threats, and one of the most significant worldwide is hop latent viroid, or HLVD. This viroid brings a host of devastating symptoms: reduced vigor, poor rooting, lower cannabinoid production, drastically reduced yield, and stunted growth. It's basically a silent killer for commercial crops. But tissue culture offers a remarkable solution to this widespread problem. That solution is called Meristem culture. The meristem is the actively growing tip of a plant. What's absolutely incredible about this specific tissue is that pathogens like HLVD frequently struggle to invade it simply because the cells are dividing so rapidly. Because of this, scientists can isolate an extremely small piece of this pristine meristem, place it in our sterile basal media, and regenerate a completely clean plant. It acts as a way to essentially cure infected genetics that would otherwise be entirely lost to the industry forever. And beyond curing disease, we have G for germplasm preservation. Cannabis genetics are surprisingly fragile in the real world. Mother plants age out, facilities suddenly close down, diseases inevitably spread, and carefully curated breeding programs can just disappear overnight. Germplasm preservation solves this by storing unique genetics in a slow growth state. Think about this. Some highly optimized collections can preserve hundreds of cultivars in a space that would normally only hold a few mother plants. It is the ultimate genetic insurance policy for both breeders and commercial cultivators. Let's move to Section 5, hardening off the plants, returning to the real world. Eventually, our tiny, perfectly preserved, completely disease-free plant has to leave its pampered laboratory life and return to the grit and reality of a normal grow space. This brings us to H for hardening off, also known as acclimatization. Now, this transition must be highly, highly gradual. These plants have essentially been living in a 5-star resort with room service, constant nutrients, zero airflow, high humidity, perfectly sterile conditions. If you throw them straight into a bright, chaotic greenhouse, the shock will outright kill them. You have to slowly reduce the humidity, carefully increase the light intensity over time, and let the roots physically adapt from a sterile gel back into a natural, gritty growing medium. It's a frequently overlooked step, but it is the ultimate test that determines if the plant actually survives. And there you have it, the complete ABCs. We covered aseptic technique, basal media, cloning, disease elimination, explants, growth regulators, germplasm preservation, and hardening off. What started off sounding like intimidating, highly complex medical lab science is actually just a logical, highly effective set of tools. Because tissue culture isn't just about growing plants in a lab. It is about preserving the past, fiercely protecting the present, and building the very future of plant genetics, which leaves me with one final exciting thought for you today. The next legendary cannabis cultivar might not be growing out in a massive greenhouse right now. It might be sitting quietly inside a tiny, perfectly sterile glass vessel, just waiting for its chance to completely change the industry. So what's in yours? Thank you so much for exploring this explainer with me today. See you next time.